Good evening, afternoon, everyone. Not too many, but I'm very happy to see all of you, those who made it. So I'll start before introducing everything. I'll just give a housekeeping announcement and say that we are the only one that's separating you from dinner, which will take place at 7 o'clock in Building 25, which is the Agriculture Institute. Okay, the French Associate, how it is exactly called, the French Associates Institute for Agriculture and Biotechnology in Drylands. So, and it's on your map, the program, Building 25. So that's where dinner is going to be. My name is Amit, and I'm a researcher at the Zuckerberg Institute for Water Research, heading this session. And I'm really excited to host our speakers. You know, sanitation is probably one of the greatest challenges in the world, and so many millions of people without proper water and sanitation solutions. So we're just gonna hear a little bit about that in this session. And without further ado, I would like to invite Professor Erika Hartmann, who is an engineer and microbiologist at Northwestern University, USA. And you'll be talking about antimicrobials and their impacts on microbes in both indoor and outdoors environments. Thanks, Erika.
All right. So let's see, I do this. Hello. Does this sound good? Okay, great. Well, thank you very much, Amit, for the invitation to be here, and thank you to everyone who's here. I think this topic may be a little different than some of the other things that are covered in this session and elsewhere in the conference, but I would like to take a slight detour and talk a little bit about indoor environmental quality and to focus a little bit on antimicrobials. So the built environment, indoor environments, the room that we're in right now, is not sterile. And I think often we think about built environments as sort of static structures, but they are in fact not. And especially we, as essentially giant walking microbial bioreactors, carry our microbes with us everywhere we go. So we have microbes in our gut, in our skin, in our mouths, and everywhere we go, we are shedding those microbes. And that has really important implications, obviously, for infectious disease and human health. But it's important to remember that not all microbes are, in fact, pathogens. Not everything will make you sick. And some of those microbes are, in fact, critically important for helping you digest your food and maintain a healthy immune system, among other things. But nevertheless, we are very concerned, obviously, about the spread of infectious disease, and so our built environments that we inhabit for most of our lives are heavily sanitized. And so one of the ways in which we do this is we use things like antibacterial hand soaps. And if I can impart just one message to you today, it is that you do not need to get antibacterial hand soap, that regular hand soap is just as effective as antibacterial hand soap and is in fact better for all of the reasons that I'm about to talk about. But hand soaps aren't the only antibacterial products that we find in our built environments. So I'm highlighting here Ticonderoga pencils protected by Microban. This is a pencil that actually has an antimicrobial embedded into the paint that goes around the pencil. Don't ask me why. You can also find antimicrobials in other personal care products like toothpastes, a lot of plastic goods, high-touch plastic goods. In this case, this is a keyboard, but you'll find especially things like baby toys or kitchen products tend to have things like antimicrobials embedded in them. And carpets, floors, tiles, all of these elements that we actually build our buildings out of can also have antimicrobials embedded in these products. So it's not just your soaps and your personal care products that I think a lot of us might think about when we think about antimicrobials, but also some of the durable goods and the very materials that we use to build with. And this has really important implications because we might make a distinction between antibiotics and antimicrobials, and in this case, the distinction that I might use is that antibiotics are medicines used for humans or animals, whereas antimicrobials are agents used to disinfect surfaces and eliminate bacteria. But in fact, bacteria don't always make this distinction, and in fact, what they see is a stressor, and they may react the same way to both of these. So what's very concerning is where we see, for example, cross-resistance between something like an antimicrobial in hand soap and something like an antibiotic that is critical for saving lives. Another key difference, sort of a functional difference for us between antibiotics and antimicrobials is that antibiotics tend to be administered for a very short period of time at a very high dose, whereas many antimicrobials are actually designed to be long-lasting and to leave residues that continue to be active in the environment. So microbiomes of the built environment, the microbial communities that surround us everywhere we are shaped by human activity and a variety of selective pressures. So one of the selective pressures that I think is very interesting is obviously the chemicals that we introduce into our environments. So I think typically when we think about chemical contamination of the environment, we're thinking outdoor chemical contamination. But one of the things that I think is very interesting is that a lot of the chemicals that we're concerned about outdoors, and I will get to that, actually originate indoors. And so we can think about the indoor built environment much in the same way, where we can actually follow some of these chemical contaminations and chemical exposures. And I'll tell a couple of short vignettes about two antimicrobials that I think are very interesting. One is triclosan, which I'm showing in orange here, and the other is benzalkonium chloride, which is a family of antimicrobials that I'm showing in blue. And whether we're talking about triclosan or benzalkonium chloride or any of a number of other antimicrobials, one thing that we find over and over again is that eventually, with enough time and enough exposure, antimicrobials lead to resistance. So everywhere we're using these antimicrobials in our built environments, there's an opportunity for the microbes that surround us to encounter those antimicrobials, and where they encounter those antimicrobials, they may develop resistance. And resistance can take many different forms. So what I'm showing here on the bottom is transformation. I think this is a particularly interesting one from a sort of bacterial physiology and metabolism standpoint. But perhaps more concerning are the others. So in the middle, what I'm showing is sporulation. So there are a couple of different strategies that microbes use that are very general stress response strategies. Sporulation is one that's particularly interesting because bacteria then form these essentially impenetrable coats and can persist for a long time in the face of a lot of stressors, including antimicrobials. And another, which I'm showing on the top, is efflux. And this is a particularly concerning form of resistance because a lot of efflux mechanisms are not terribly specific. And so something that can efflux an antimicrobial may also be able to efflux a clinically relevant antibiotic. And antibiotic resistance, just for context, is a massive global public health challenge. I think everyone is obviously aware of the COVID-19 pandemic, but the antimicrobial resistance it's sometimes called the silent pandemic. So there were something like 14 million deaths due to infectious disease in 2019. About half of those were due to only 5 pathogens, and those are the escape pathogens that we're very, very concerned about antimicrobial resistance in these organisms. So these are infections that we used to be able to treat routinely with regular antibiotics, and now we're finding that our ability to treat these infections is vastly diminished, and we only expect that this will increase. So I invite you going forward to think about the built environment not as the static structure that we inhabit, but actually as a dynamic chemical and biological system where we have chemicals that are coming in from both indoor and outdoor sources, such as the personal care products that we use and also the building materials, and that those chemicals are partitioning and moving around and encountering the built environment microbiome, including the microbes that we bring in with us everywhere. So one of the questions that I have tried to address through my research is resistance in microbiomes of the built environment actually shaped by antimicrobial usage? We know that this is the case where we have, for example, exposure to clinically relevant antibiotics in the human gut. What that results in is restructuring of the microbial community, so different microbes are left after an antibiotic treatment, and those that are left tend to be enriched for antibiotic resistance. We also know this is the case in wastewater treatment, which is also a giant bioreactor. So in this case, what we're looking at is the number of gene copies of a particular, in this case, efflux pump. And if we perturb this system with triclosan, in just over 2 weeks, we see over an order of magnitude increase in this particular efflux pump. So what I wanted to focus on here is triclosan, which is an antimicrobial that was commonly found in toothpaste and other goods. It was actually banned in the United States from consumer hand washes in 2016 and has been largely phased out. But it was an enormously high production volume chemical, and as such, it is ubiquitous in indoor dust. So just showing that there's an enormous amount of this chemical in indoor settings. Exposure of microbes to triclosan is linked to antibiotic efflux, which is that particular resistance mechanism that I mentioned earlier. It's also linked to horizontal transfer of mobile antibiotic resistance genes, and this is sort of a general concern for emergence of new resistance strategies in the future. And so to look at the role of triclosan in enriching for antibiotic resistance in indoor environments, we actually ran around and collected a bunch of dust samples from athletic facilities. And we extracted triclosan and used a specific quantification method to identify and quantify triclosan. We also identified antibiotic resistance genes based on what we know about, in general, antibiotic resistance genes. And we also took a culture-based approach, so taking a very tried and true, like 150-year-old approach to microbiology. And what we found is that in dust samples that had higher concentrations of triclosan, we tended to find more organisms that had more resistance genes in their pangenomes. So what this basically means is that where we have more antimicrobials, we have microbes that are not just potentially resistant to those antimicrobials, but resistant to a wider variety of things. So many organisms tend to have more than one resistance strategy, and when you enrich for organisms that are resistant to one thing, you're potentially enriching for organisms that are resistant to a plethora of things. And we also found that buildings that tended, again, to have more triclosan in them tended to yield more isolates that were actually resistant to, in this case, we tested clarithromycin, ampicillin, and tetracycline, so 3 different families of antibiotics. And this is important because it shows that we can actually grow things that are actually demonstrating these resistance strategies. So it's not just, oh, this is a bunch of stuff that's in dust, but everything that is in dust is dead. It is, in fact, viable and culturable. So this is an interesting association that where we find more antimicrobials, we find more resistance, but is this, in fact, active selection? Is this, in fact, something that is actually ongoing? And so to answer this question, we switched tracks a little bit. Again, I mentioned that triclosan was banned in the United States and has largely been phased out in favor of benzalkonium chloride. Benzalkonium chloride, incidentally, usage has skyrocketed over the last couple of years. First, in response to the ban in the United States, but then second, massively in response to a huge uptick in cleaning practices due to the COVID-19 pandemic, and a lot of surface disinfectants now having benzalkonium chloride in it. So benzalkonium chloride is the replacement for triclosan in antibacterial hand soap in the United States. One of the things that we know about benzalkonium chloride is that it's ineffective against spores. And much like triclosan, exposure to benzalkonium chloride is linked to an increase in both antibiotic transformation and efflux. And we didn't actually look at soaps in this particular study. I was really interested in the world's first microbicidal paint. So this is actually a latex paint that you could get to paint on your walls. I'm not sure why you would want a microbicidal latex paint on your walls, but this is a thing that you can now get. And this is registered with the US EPA, where they tested 5 different pathogens and showed 3-log removal after exposure to these surfaces. And we compared that to a conventional mold-resistant paint. So most paints, whether or not they're labeled as antimicrobial, actually contain an antimicrobial. And this is to protect the paint while it's in the can, to actually keep the liquid paint from growing mold. And because we don't know what that antimicrobial is, we wanted a third sort of comparison group. So in this case, we used a clay paint. This is an all-natural, no added chemical biocides product. It's sort of the extreme end of the eco-friendly product spectrum. And we took from the isolates that we were actually able to grow from the dust samples that I mentioned in the previous study and created a mock community that was representative of the microbes that we would typically find in a built environment. And we exposed these bacteria to surfaces painted with these 3 paints. And lo and behold, what we found is that actually whenever you put bacteria on a dry surface where there's no water, it's cold, there's nothing for them to eat, they basically die whether the surface has an added antimicrobial or not. However, what you're seeing here is about a 3 to 4 order of magnitude decrease in viable bacteria, but it doesn't go all the way to 0. There are some things that persist. And the bacteria that persist change depending on the environmental conditions. So here we're looking at 3 different humidity conditions and the clay versus the mold-resistant paint. And these are the 5 different bacteria that we included in our community. And when we look at the microbicidal paint, which I'm showing here, we see overwhelmingly that Bacillus timonensis is the organism that takes over in these microbial communities. So for anyone who's familiar with Bacillus, you might know that one of the hallmarks of these species is that they like to form spores, and I already mentioned that benzalkonium chloride is ineffective against spores. So we thought that they might be surviving by forming spores. And so we actually took some images. These are electron microscopy images, and I'm showing the vegetative and spore form of our Bacillus timonensis on the top. And then these are images from samples that we actually collected off our surfaces, and we see both vegetative and spore forms. So there's something in addition to spore formation that's actually allowing these bacillus to persist on these surfaces, which we thought was pretty interesting. But basically, to me, the take-home message of this research is that where we see more antimicrobials, we tend to see more resistance, and where we apply microbes to an antimicrobial treated surface, we're actually selecting for microbes that have different resistance strategies. So then one of the really important questions that we can start to ask is about the fate of benzalkonium chloride in the environment. So as I mentioned, these chemicals are used extensively in personal care products and also in building materials. So where do they actually go? Since we know they're not staying in those personal care products or materials, where do they actually wind up? And so we performed a systematic review and looked at hospital effluents, we looked in wastewater treatment, we looked at wastewater effluent, and we're finding that benzalkonium chloride is absolutely being passed through, for example, in hospitals where they're used extensively for disinfection, and winding up in biocides, winding up in wastewater effluents, and winding up then in receiving bodies of water. So all of the problems that I just mentioned for indoor environments are also potentially happening in outdoor environments. And this is really critical in the One Health framework, so this idea that human health and environmental health are intrinsically linked. And so the more antimicrobials we put out into the environment, especially knowing that these antimicrobials are designed to leave long-lasting residues, and so they're actually designed to persist in the environment, is potentially just creating an opportunity for more and more resistance to develop. So we have done some work looking at antimicrobials and their impact on gray water reuse, which I'm going to let Zeev talk about just after me. So I'll sort of end my little research spiel there, and just encourage you to think about microbe-chemical interactions. So again, the idea that the environment around us is actually a dynamic biological and chemical system, and that the choices that we make with our personal care products and our building materials impact both indoor and outdoor environments, and potentially have huge sort of long-lasting impacts on global phenomena like antimicrobial resistance. So obviously I didn't do any of this work alone, so massive thanks to my wonderful students, and we are recruiting, so if you or anyone you know is interested in antimicrobial resistance, I would love to talk to you. Chicago is a lovely place in the summer. Also in the winter, but for different reasons. And of course, also thank you to all of our funding agencies, and I'll take any questions. Yeah, so that's a great question. I'll just repeat it. Is there a way out of this vicious circle of we use antimicrobials, the bugs develop resistance, so we have to come up with more antimicrobials and they develop more resistance? So in theory, antimicrobial resistance should be an expensive trait for microbes to maintain. And so if that selective pressure is removed, in theory, the resistance should disappear. In practice, that's not actually what we see, and I think that's really, really interesting, and I have some work going on actually in Pakistan looking at ESBL-producing Enterobacteriaceae and trying to understand what ecological advantage they have for maintaining these genes, even in the absence of antibiotic pressure. What I really think is necessary is in the same way that we're talking about antibiotic stewardship in the clinic, so thinking about not prescribing antibiotics unless you know someone has a bacterial infection and trying to do the sort of minimum dosing, that we need to think about that also in a cleaning context and start to ask the question of, do I actually need to use a disinfectant here? Is there an alternative strategy? Am I good to clean with just a mild detergent, which, especially for household applications, is better for you because then you're not exposing yourself to a disinfectant, and better for, you know, your septic system or the downstream wastewater treatment plant or whatever. So I think it's a paired approach of understanding the ecology and why certain bacteria might maintain these resistance traits, and then also just reducing our use of disinfectants in the same way that we're tailoring our use of antibiotics in the clinic.
Ask about first part of your talk when you investigate dust in spore facility, and I guess so. Do you think accumulation? So do you observe accumulation of resistant genes, or it's reflecting from outside dust?
Yeah. So that's a great question. So do we see accumulation of antimicrobial resistance genes? What's going on basically in dust? The short answer is I don't know. So that was a one-time study. We basically have one time point. What I think is really interesting is that time point was prior to the ban on triclosan in the United States. So in theory, we could go back now and collect samples again and see if there's any changes, both in the triclosan content of the dust and the antimicrobial resistance of the dust microbial communities. Dust is sort of like, it's the indoor air equivalent of like a sediment core or an ice core. And so in theory, what you're seeing is the result of accumulation over however long the dust has been sitting there. It's also possible that what it's reflecting isn't necessarily processes that are occurring in the dust, but processes that are occurring in the room. So it might not be that triclosan is in the dust and the microbes are in the dust, but that actually triclosan is on my skin. And so what's happening is happening on my skin, and then everything is sloughing off of my skin into the dust. We haven't had an opportunity to pursue those research questions yet, but I would absolutely love to, and I think they're very interesting. All right, thank you.
So thanks, Erica. And the next presentation, and that's a bit of a change from the program because it just makes more sense, would be given by Professor Zeev Ronen from here, from the Zuckerberg Institute for Water Research. And he'll be talking about the reasons, are there reasons for concern for antimicrobial resistance in gray water?
Good evening, everybody. Thank you for the introduction. And I would like also to thank Erica that actually lay the foundation for my talk. And what I'm going to talk with you in the next 20 minutes is a topic that we are following in the last few years. And this is the problem of greywater quality. And we will start with the reason for using greywater, and the main reason that we are starting to use more and more wastewater, particularly because the lack of water resources around our houses, and sometimes also because of the lack of sanitation. So reusing of wastewater for non-potable applications like irrigation, toilet flushing, and even the hand washing after treatment can be a water-saving approach under changing climate, where we have less water for non-potable application, and therefore the idea of using greywater is a great idea. And in general, domestic water from baths, laundry, showers, and sinks can be reused for various purposes, including toilet flushing, garden and landscape irrigation. And what we need to know is that between 50 to 80% of the residential wastewater is gray water, and it's coming from, of course, from non-toileted sources, and it's coming from kitchen, showers, sink, and also washing machine. And clearly, Eric already presented that using all of the cleaning products during these activities can severely affect the greywater chemical properties. Okay. And so we can divide the greywater into light and dark greywater. And very often, one of the problems that we are facing is that we are using greywater on site. We are not using a centralized wastewater treatment plant, but we are using a on situ system for treatment of the greywater, and therefore we prefer to try to deal with the light greywater that coming from the bathroom or from washing machine. And obviously, like already mentioned, it contains all kinds of chemicals and all kinds of materials that need to be treated before we can reuse the wastewater. So the reason for greywater treatment is coming not only is that we want to save or reuse water, but also from the fact that in Israel and all around the world, there are people who are lacking sanitation facility, and mostly it's actually collecting wastewater. So for instance, we have a settlement not far from here. It's a Bedouin settlement that is near Beersheba. It's called Umbatim. It's about 1 kilometer from a central wastewater treatment plant, but it's lacking a sewage collection system for various reasons. And so toilet waste is kept or collected in campsite or all kind of improvised system. But greywater, for instance, is just discharged to the environment, and therefore to protect the environment and protect people who are living near the discharge points, we need to do treatment. And clearly, if we don't do treatment, we can affect negatively the environment and human health. So the main problem is chemical contaminants, but also we have to consider that the greywater contain biological, microbiological contaminants, and the problem is not only in Israel. The problem is all around, especially in places where we have arid environment. You can see here a recent review about global gray water treatment and reuse research. Leading countries are in doing this research is Australia, the US, China, India. But important thing is that Israel is leading in this research. When I look to the details of this research, both Amit and Eran Friedler from the Technions are the leading researcher in the topic. So the problem of gray water use, it's not locally, it's also a global problem. In Israel, for a very long time, the group of Amit was doing research on treatment of gray water, and we were using the recycled vertical flow constructed wetland for treating gray water, and the system is quite simple. You collect the gray water, let sediment to settle in the bottom, then the raw gray water is discharged on the surface of a filter bed. The water is percolated into a lower container where it gets aerated and then recycled a few times until you get the right quality of the effluent. When you get the right quality, you can use it for irrigation. Now, the water before it's discharged can be good under filtration or get disinfection, but we know that in most cases these systems are not so effective. So in many of the places that using this system, either the filtration or the disinfection units are disconnected. However, the treatment is very effective. Well, like we can see here, that the major contaminants are removed. And also, if we look on the effect of irrigating the soil with treated gray water, we see that it's quite effective, and there are not many incidents of presence of fecal coliforms, which are the benchmark for microbial pollution of the soil. Nevertheless, although we know that the gray water treatment is effective, we don't know anything about emerging pollutants that we learn more and more about, like micropollutants, microplastics, antibiotic-resistant bacteria, and also antibiotic-resistant genes. So we aim now to look on this system and to understand whether they remove these pollutants, particularly antibiotic-resistant gene and antibiotic-resistant bacteria. So our study is dealing with of course, with antimicrobial resistance, and already Erika presented and explained what is antibiotic-resistant, microbial antibiotic resistance. And beside the health problem, there is economic cost. And if you have to treat a patient that is suffering with MRSA, which is a metacycline-resistant Staphylococcus aureus, it's double than the cost of treating a patient that is suffering from sensitive organisms. So there is a strong burden on the health system treating antibiotic resistance in the hospital. And this already been shown, but I would like just to draw your attention that the predictions that in 2050, deaths from AMR or from antibiotic resistance will be equal or larger than any other diseases like cancers and other problems. And we already, again, I'm repeating myself, but Erika presented the mechanisms where bacteria defend themselves from from antibiotics, including enzymatic destruction of the drug or prevention from the drug entering into the cell, or a modification of the drug target, or rapid ejection of the drug, which is the efflux pumps that you just said. Now, what are the mechanisms that human can be exposed for antibiotic-resistant bacteria, antibiotic-resistant gene from wastewater use? So clearly, if the antibiotic-resistant bacteria that are present in the effluents, whether it's just wastewater or gray water, get into the wastewater treatment plant or the on-site gray water treatment, are not removed, they can discharge into the environment. Then there are two possibilities. First, direct exposures of animals, human, from the water that is discharged into the environment or used for irrigation. And secondly, indirect route where the genes of the antibiotic-resistant bacteria is transferred to environmental bacteria that acquire the resistant, and then especially when we deal with irrigation, it can be transferred to plants, and then we are exposed to these genes and resistant bacteria. However, 5 years ago, there was a paper in Environmental Science and Technology by leading water quality experts, including Charles Gerber from Arizona. And they questioned the notion that there is a risk from antibiotic-resistant bacteria, especially using treated effluent for irrigation. And they claim that there is no enhanced environmental antibiotic resistance. Usually what we see, it's transient and there is no risk. And this is something that been shown for quite a few times with irrigating with wastewater treatment, treated municipal wastewater water, but not with gray water. So we study what we have in the gray water, what we have in the raw gray water, what we have after the treatment in the recycled vertical flow constructed wetland, and then also what we have in the irrigated soil. And importantly, the systems are located in households, and most of them are located, what we studied, the systems are located here in the Midrasha, and we study these systems for quite many years, and we try to answer this question. First of all, do we have antibiotic-resistant bacteria? Do the system remove them? Do they accumulate in the soil? Okay, so let's go to our results. The first thing that we have done is quite simple. Just take water from before and after treatment and put them on plate, traditional microbiology. In this case, we use amoxicillin, which is one of the first antibiotics that the doctor will prescribe for any infection. And we just took water samples from 3 different households. What we see, we use 3 different concentration and control before and after, and to our surprise, we have seen quite a bit of resistance. The resistance for amoxicillin is defined by the minimal concentration affecting or inhibiting the growth of bacteria, and usually it's around 20 milligram per liter or even less. So we see that we have quite a large number of resistant bacteria, but what we can also see is that the system is quite effective in removing between 99 and sometimes even in higher efficiency, even 99.99. So we see that the system is removing, but still they are present in the treated gray water. Now, Daniela was actually trying, in addition for looking for the antibiotic-resistant bacteria, to look for the genes. And what we see here is a set of genes, including genes with resistance to triclosan, but to other antibiotics. And in addition, for looking on the resistance in the raw and the treated water, she was also looking on the community or the number of resistant bacteria in the filter bed of the system. And it's not surprised that the filter bed also contained quite a big community or number of antibiotic-resistant genes, since this is the best place for them to stay. But what we see is that we also see that the treatment sometimes remove, especially genes that are coding for beta-lactamases, but sometimes it's not as effective, like if we look on the INT1 or integron 1, that is a representative of horizontal gene transfer in bacteria. So we see we have the antibiotic-resistant bacteria in the water, in the treated water, and we have also the genes. And with the help of Erica and her postdoc, we also look for the families of antibiotic-resistant genes, and surprise, surprise, we get the effluent pumps dominating in all of our samples, whether they are in the raw or whether in the treated gray water, meaning that there is something probably related not only maybe to residue of antibiotics, but to the chemicals that we are using in our household. Later on, we look for the abundance of antibiotic-resistant bacteria in irrigated soil. And again, fortunately for the design from Amit research, each household contained both treated gray water irrigated plot adjacent to a freshwater irrigated plot. And what is the main surprise that we see that if we look on the antibiotic-resistant bacteria, here we are talking about resistance to amoxicillin, kanamycin, and cipro, and we see that there are not much differences. Okay, and this is a really interesting point because it can come from different reasons. One, that maybe since the plots are so close together, maybe we have kind of contamination, movement of animals through the plots that contribute to the distribution of the resistant genes, or alternatively, that actually we are what we see in the soil, it's something natural, and it's not coming from the gray water. So this question still need to be addressed, and again, the same question, the same results on antibiotic-resistant genes now is present in fresh water and treated gray water irrigated soil. And what we see is that the only very significant difference that we can see is the INT1. And usually the INT1 gene is represents a potential for horizontal transfer of resistant genes from the irrigating the water to the soil. So the other genes look that are there are no differences, and maybe again this is because either that this resistance is natural, or maybe it's a contamination between the plots. I will switch gear and we try to understand who are the organisms that are present in the gray water in terms of resistance. And we actually, all of these bacteria, all of these isolates are resistant for different antibiotics, amoxicillin, kanamycin, Cipro, tetracycline, and also ESBLs, which are extended spectrum beta-lactamase containing bacteria, which is our most concern for us because ESBL bacteria are resistant to also third generation beta-lactam antibiotics, and this is really alarming. And the main thing that we have seen is that most of the bacteria are protobacteria, or more specifically, gamma protobacteria. And this is of concern because usually we're talking gamma protobacteria, either Enterobacteriaceae or Pseudomonaceae, which are, some of them are facultative pathogen. Looking on multiple resistance in the isolates, we can see that some of the isolates are containing high resistance for multiple antibiotics, but there is no real pattern that we can deduct from this table, but still there is a potential that the antibiotic-resistant bacteria are multi-drug resistant. Importantly, the question if we can do something about it, and one of the answers is yes, we can keep the disinfection unit operating. And this is just an illustration of exposing different resistant bacteria to chlorine, and we can see that given that we have enough time, we can reduce the colony forming unit in the water to below detection limit, but sometime for some of the strains it will take longer. Looking on the community of the bacteria that is present in the water, both the treated and the raw, in the different households, again, what is jumping to our mind is that the dominating organisms are protobacteria, and when we are trying to look deeper, again, what we see, the dominating is a gamma protobacteria consisting of Pseudomonas and Enterobacteriaceae, which are facultative pathogens. We also see quite a bit of epsilon protobacteria that we don't know that they are really have any health impact. Okay, so to conclude, we have antibiotic-resistant bacteria in gray water. We know that their origin is not in potable water. It's not coming from the supply water. We check it. So it's related to what we are using in the household, the chemicals that we are using in the household. Probably these are the main drivers for the evolution of resistance. The treatment is effective, and we see that the treatment removes both bacteria and genes. But if we want to give more protection to us and to the environment, we should apply also a disinfection to the treated water. We see that there is no significant difference between gray water irrigated soil and freshwater irrigated soil. And this is still a question that we need to understand why there is no difference. And there are a lot of studies that are trying or claiming that the soil microbiome is quite resistant for invasion of bacteria from the outside, whether it's from the gray water or municipal treated wastewater. And there is a lot of debate about this issue. We shows that we have a lot of facultative pathogen, but the question here since we use culture-based methods, the question was we just had a cultivation bias, and we should better look more deeply into the microbiome, to metagenome, to understand who are the real bacteria that are persist in the raw, treated, and irrigated water. We have seen no change in the community due to irrigation, and this is not something new. We are not the first to show that. But my last conclusion is that we still need to have a definitive quantitative microbial risk assessment to do on irrigation of gray water, in particular in relation to antibiotic-resistant bacteria, antibiotic-resistant genes, and maybe also to the drivers of this resistance, the micropollutants, maybe the microplastics, and others. So I would like just to thank the founders, the Tzuk Maccabi Foundation, that actually found this researchers that he was granted to Amit. And in addition, I would like to thank the BGU Northwestern University SING grant that provided for me and for Erica. And this research was also supported by this seed money. And with that, I would like to thank you and entertain any questions that you may have. Daniela.
You said in order to remove antibiotic-resistant bacteria, there should be an additional disinfection. So in the long term, would it not be an additional specimen bacteria and increase the antibiotic-resistant in the system?
So the other system that we tested and you sampled, all the disinfection units are not working over there. There is no disinfection currently, and the disinfection should happen before irrigation. So the disinfection so before you release the gray water to the environment, you should treat with additional disinfection. But here come additional question: what will happen after disinfection? We will have disinfection byproducts, and maybe these disinfection byproducts will actually promote maybe microbial resistant in the receiving soil. We don't know. So there are still, we need to balance between the risk and the operation of the system. And this is why we need to do some kind of quantitative microbial risk assessment. Maybe our results, if we will do some accurate risk assessment, maybe we can stay without disinfection. But still, we have to think about it in order to minimize the transfer of resistant bacteria and resistant genes to the environment.
Thank you, Zeev. And now we will be moving to the third presentation that will be given by Professor Hadas Mamane. She's a water treatment and engineer at the University of Tel Aviv University. And Hadas will talk. Hadas actually was also researching a lot in India and in recent years. And she's going to present treatment of sewage ponds in rural India without energy investment using the coupled microalgae bacteria biofilm technology. So it will be combined with microbial or bacterial treatment now, and then we'll be moving to other methodologies of treating or producing sanitation. Please, Hadas. And I guess that's the way it works.
Hi, everyone. Thank you very much for all those brave people that are still here. So I would like to talk about work that I'm doing in India, in rural India. So we know about the problems. Actually, the European Union has set a goal of achieving zero pollution for a non-toxic environment by 2050. However, there's a huge question: can we really achieve a non-toxic environment? We know that the conventional wastewater treatment plants, like activated sludge, are highly effective and also they're efficient at removing organic matter and nutrients. However, they are expensive and energy demanding. And if we're talking about low and middle income countries, they cannot actually implement these technologies, especially in rural areas, peri-urban settings, slums, and environments like that. In addition to that, 360 cubic meter of wastewater is produced per year. Only 10% of it is treated in those conventional wastewater treatment plants and reused. And the rest of the wastewater is actually either non-treated or somewhat treated and dumped to the environment. Therefore, we have a lot of wastewater in the environment, and we all know about the problems that this wastewater results in pathogens, release of greenhouse gases, impact the human health, societal issues, and so on and so forth. Also impact the amount of days that sickness of children and stuff like that. So these are just a few pictures from sewage ponds that we're working on rural areas in India. And we can see that actually a sewage pond is a sewage pond. The sewage is actually discharged into these like huge ponds, which are about like 2.5 meters deep. And we have growth of algae and plants, and there's also waste within these ponds. So these ponds are actually, I would say, in many cases are very contaminated, and usually these ponds, the water from these ponds is pumped into agriculture irrigation. So there's no treatment for most of these ponds. It's an environmental hazard. In some cases, we have overflow. There is infiltration to the groundwater because these ponds do not have a layer that actually protects them from infiltration to the groundwater. They're used for irrigation in many cases, and they result in soil damage, health damage, and whatever type of damage. And these are the cases that actually the sewage is collected into the ponds. In other cases, there are issues where the sewage is even not collected into a central location. So our goal is actually to scale down, localize, and decentralize systems for rural and urban areas with an economic benefit, and actually trying to mimic natural consortiums of macroalgae and bacteria that anyway occur indigenous into the sewage ponds. And these advantages actually inspired us to develop a process that we termed the attached coupled microalgal bacteria biofilm, or CMBB, which is an alternative for sewage treatment, which can actually provide sustainable food and also biofuel generation upon collection of those algae. So the concept is very simple, and we examined it initially using sponges, like the regular sponges that we take a shower. On these sponges, we know that bacteria or biofilms, they have a tendency to attach to surfaces. So whenever we have a surface and water, we'll have a biofilm forming. And within these sponges, the bacteria and the algae can actually attach to them. And then we have a process where we have biodegradation of organic matter by the microorganisms to release carbon dioxide that is actually used by the algae to generate in situ oxygen without actually aerating these ponds, because one of the issues is that about 30% of the cost of activated sludge processes is the aeration, which obviously, and of course, they don't have the ability to use aeration processes, and therefore this is an in situ low-cost aeration. These are some of the people that we work with in our process in India. By the way, in the center is Sichawal. He's a guru. He actually is someone that did pretty amazing things in the Punjab area, where he actually cleaned many villages and nalas and channels from waste and wastewater. So our initial model was using a sponge support, and we tried to actually, at the beginning, we tested our study in lab setting. So we did like very simple study. We took sewage, effluents, and all kinds of variations of different treatments. We actually dumped sponges, and we tried to follow the process. So in one of the studies, we wanted to know, for example, can these sponges actually remove micropollutants? In this case, for example, we saw that the microalgae, actually chlorella was able to remove 40 to 50% of ioxol following incubation. And we also examined, we paralleled that with examining ozonation processes. So we saw that we have the major pathways were oxidation and hydrolysis. The next step was actually to use sewage. And here, in this case, we tracked different micropollutants, and we saw that they were able to degrade also micropollutants using the CMBB process. In addition to that, of course, we were able to remove ammonia, BOD, and COD, and all the standard water quality parameters. In addition to that, we examined the impact of these treatments on the different subgroups. So, for example, we can see that there is a change in the communities following the CMBB process, and because these are actually organisms that are attached to biofilms. Post both of your talks, I was just thinking, how are the antibiotic-resistant bacteria in biofilms compared to free-floating in wastewater? So just kind of like after your talk, it kind of like interests me to know if we are selecting in biofilms more antibiotic-resistant genes. So I don't know, this just came up post these two talks that were in the beginning. So this is just to show that we actually are forming flocks on these sponges. And this is the scheme of the process that we're doing. So basically, it's a very simple treatment where we're taking the sewage that goes into these ponds, but before that, we're removing very roughly with bars, just whatever we can separate. Then we have also a floating device where we can remove whatever floats in the sewage, and then we have the ponds. Within the ponds, we're actually adding those sponges, and then we're using the water for irrigation. At some point, we are collecting the sponges, separating the algae for biofuel production, and we're also adding low-cost sensors to this system. So this is kind of the entire scheme where we're going to. However, sponges are made of polymers. Polymers are made of fossil fuels, and definitely we don't want to dump sponges into the environment. In addition to that, they're non-biodegradable. So we thought, okay, how can we actually? So we saw that the sponges work very well. It's a very simple technology. We just put those sponges into these ponds. They float on the water, so we chose sponges that can float on the water, and they were able to improve the treatment. So we thought, okay, what can we do different because sponges are non-biodegradable? So we decided to test different types of supports, and we kind of came out with the idea that is called the mushroom. And this is kind of like where our vision came. So you know that like with the mushroom, you have the cap and you have the leg. So within the cap, we have air, so these supports can actually float on the water. We don't want them to settle. We want them to float on the water. And then the leg is actually an area with the has a high surface area. We tested different sponges. These are, for example, 3D printed with PLA. Polylactic acid, which is biodegradable in about like a year. It takes 1 year to biodegrade these carriers. And they just float on the water like you see in these pictures. And they are actually a zone which enhances bacterial and algae association, which generates in situ oxygenation and improves the quality of the effluent before irrigation. We tested different configurations, different designs. We tested the cube. We tested spheres. We tested with different infills to understand honeycomb design and different designs to understand which design would we enhance the most amount of biofilm within these designs. And at the end, we decided to work with a mushroom type of support. Before we actually dump these supports into the wastewater, we actually do an enrichment process. And what we saw is that if we pre-enrich those supports with the biofilm, then they actually oxidize the ammonia, remove the COD and the TSS and everything much faster compared to non-enriched biofilms. So here we actually we developed a reactor where we put all these supports. We enrich the biofilm in a short process, and then we separate them and move them to the ponds. And this is, for example, one of the pilot scale studies that we did. So we moved from the lab to a pilot study. This is with 7 cubic meter of sewage. And it has kind of like the depth of a sewage pond, which is about 2.5 meters. So this 1.5 meter deep, and the retention time was about 7 to 12 days. This is just a few graphics to show that actually we had total COD and soluble COD removal with the supports. We also tested supports with different infill, and then actually we used this water for agriculture irrigation. We're now collecting data from the field with using actually wheat. We also want to move to rice post-wheat. And we're trying to understand what is the difference between irrigating these crops with either raw sewage or treated sewage with the sponges, or the third testing that we're doing is actually we are isolating different algae from different zones in the Punjab area and trying to see which one works better in agriculture irrigation. And this is just a picture of one of the different isolates with the algae. However, I just want to show a few issues that are related to wastewater treatment. As engineers, we are actually educated that if we come with a technology that would be simple enough, that will not require retrofit of existing for example, in this case, an existing pond, they don't have actually the resources to take this sewage to an activated sludge system or any other type of wastewater treatment. So we kind of like, we're thinking, if we improve the treatment of if we improve the effluent quality with a simple technology, will it actually work? And therefore, there's many questions to ask here. We need to also look not only the technical aspects, but also the societal and the economical aspects. So will farmers use this technology? Farmers are one of the stakeholders. Then we have additional stakeholders are the public. And what is the public perception towards using treated wastewater compared to the alternative? So the alternative is actually using groundwater in some of the cases. So what we did, our students were in the field for a few months now in India. They only came recently, just a few weeks ago. And they went from village to village, and they looked, for example, in which, I'll just show you. Okay, so they looked, for example, in which cases did the farmer use the sewage ponds, in which cases did they use groundwater, and they also looked at energy, which type of pumps, is it solar, diesel, and looking at the entire picture to try to develop an economic model in simulation to try to understand what's happening. So for example, we looked at different aspects of, I can't get now into all the different aspects, but we developed this simulation to look at the entire picture of using wastewater, groundwater, and treated wastewater with our technology and try to understand and compare and contrast if the farmers will use it. We also looked at the farmers' perception of using recycled water compared to groundwater and cost comparison. Just to show you like a quick, some quick results. So for example, the upper graphics, we want to understand what is the size of the rice plot and how many tubes wells do you have when you also use sewage ponds. So what we can see here is that actually when people also have ponds, they use less tube wells within their field. In addition to that, we wanted to examine, because there's a notion with the farmers when we surveyed them, that when they use sewage, they have to use less fertilizers. So we wanted to actually examine if it's correct. We're still crunching the results. I mean, this is like very initial because it's very complex. So some farmers use sewage ponds, others use ground wells. Some use both of them, so the field is kind of mixed. Others, for example, and also there's water that is actually rainwater. Therefore, it's very complex to understand and to really dive deep into the understanding of the impact of these treatment technologies. We also did assessment of the water quality in many, many ponds, and also assessment of the soil post-irrigation with sewage versus groundwater. We didn't yet analyze the data. This is like part of our team being in India. And we also like to end that we also wanted to understand the public. Will the public actually use or consume food crops that were irrigated with sewage? So we did a study where we compared, where we took, so this is actually a very interesting study. And you see here, this is irrigated with groundwater, and the other one is irrigated with treated sewage. So we also explained that this is treated sewage and not raw sewage. Okay, it's very important to do this differentiation. And we divided the counter. We went to different markets to understand what would be the public perception of eating food that was irrigated with treated sewage. And we examined different parameters, for example, weight, crop type, the pricing, and just really quickly, this is what happens when we ask the consumers, will you consume food that was irrigated with either treated sewage or groundwater? Or what happens if we reduce the cost? So if we reduce the cost, can we actually impact public perception on consuming treated wastewater crops? And what we saw is actually that when we reduce the crop, there's a higher percentage of consumers that actually consume this type of crops. And we asked why groundwater. Well, 30% thought that groundwater was healthier versus sewage. Actually, 20% of the people thought that actually consuming crops from treated sewage is more sustainable source for irrigation. So this is the sustainability aspect of using sewage. And just to conclude and end with this, when we're doing projects in these type of settings, it's not enough to examine a technology in a pilot scale. We really must understand and map the entire stakeholders that would be interested. So it's the government, it's also looking at state governance, it's also looking at the different, the consumers, the farmers, and everyone. We saw that this technology is actually very simple. No retrofit can improve the effluent, and it can be integrated also in different ways. So, for example, as a perimeter barrier or in low or high rate algae ponds with minimal energy investment. And thank you very much. No, we did these testing only in markets in the Punjab area, so we have no comparison to other countries. And what we basically saw is that when we reduced the cost, a higher percentage of people were willing to purchase the food that was irrigated with treated wastewater. But also there was, like I said, a huge percentage that thought about the sustainability aspect of using a resource such as treated wastewater, which we're very happy about. Yeah, 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 yeah.
Is there any regulation of using treated wastewater for irrigating any crop in Israel?
So he asked if there's any regulation. Well, there is regulation. There are standards for using wastewater for agriculture irrigation. However, standards set alone and reality alone, in the Punjab area, due to the climate crisis, the monsoon pattern changed completely. So they have a very short time of the monsoon, and they move from paddy to wheat. And they have a very short time for both of these crops. Therefore, there's issues with supply of water. They have to pump deeper into the wells in order to get groundwater. And that actually incentivizes farmers to use sewage as a resource for water. And it happens more than, so I tried to read about how much it happens globally. It happens more than, I don't remember the numbers, but it's huge numbers. And so these are kind of like waste stabilization ponds, but the idea is, can we manage it a little bit better to improve the quality? And also remember, always with environmental issues, it's the least of evils. So this is the alternative. What can you do to improve it? And hopefully in the future, it will improve even more.
Thank you very much.
Thank you.
So we move on for treatment or sanitation treatment. And from microbes, we're moving to thermal treatments for sanitation. And I'm happy to invite Dr. Roy Posmanik from the Volcani Center. He's in Neve Yaar Research Station. And Roy, just 1 second, Roy, defer. Roy will be talking to us about subcritical water, a green solvent for resource recovery and circular economy. Thanks, Roy.
Thank you, Amit, and thank you all for staying. Thank you for the invitation, the introduction. I'm really happy to be here. Today I will give a very short introduction to this technology called subcritical water extraction, or if you heard about hydrothermal processing, hydrothermal carbonization. All of those are the same technology, using high pressure, high temperature, and high pressure water as a solvent for a reaction. Actually, usually I'm starting all presentation with lots of slides, trying to convince that we have a problem, but this time I thought it's unnecessary. But I do want to show this map, which I saw last night for the first time, and it was very interesting to me to see how many places around the world having such a short sanitation, and all of those are actually also subjected to desertification, which is the reason why we are here. And thank you, Hadas, for giving the previous talk. And I think it's a very I'm very optimistic in terms of not just from the willingness of the society to adopt resource-oriented sanitation system, but also the need, because there is a very important need to adopt those technologies, and I think of it like the cellular phone. Most of the people living here or here may not have a line phone, but they do have a cellular phone. So they just cross the technology, and maybe just resource-oriented technology can be adopted easily on in these areas. I'm not sure, but I'm more optimistic. Another movement that we have to consider when we're talking about waste to resource, in this case biofuel, but it's not necessarily should be biofuel, is the movement to the carbon negative system. So if in the past we were mostly in let's look about on the biofuel production. It was very carbon positive. They're based on carbon positive technologies. Also the feedstocks, the technology were very not efficient, and the movement is towards the carbon neutral or even carbon negative technology, and that's why we are talking. I want to introduce the subcritical water as an optional emerging technology as one of the compartment of wastewater treatment. So if we look at the phase diagram of water, just very quickly, and just skip the boiling point of water and keep the system under pressure, we will find ourselves in a very interesting zone from the chemical perspective towards this point, what we call critical point. This is in 374 degrees and 22 megapascal. Beyond that point, we cannot recognize the state, the change in the state. It's called supercritical fluid, but let's not get there. Let's stay here at the subcritical region, and this is a very interesting region if we're talking about biomass processing. And if we go deep into those properties of water at what we call the subcritical region, we see a dramatic drop in the dielectric constant and in the density. Means that the solvent is much less polar. And at the same point, we have an increase in about 2 orders of magnitude in the ionic product of water. Means that this solvent is much more reactive. So processes that at room temperature should have been catalyzed by adding acid or base at the subcritical region are practically autocatalyzed, which makes this as a very interesting green solvent. So we're not at the field scale, or we're not in India yet. We're still at the lab scale, and in the lab it looks like that. So we have like a pressure cooker. Our own pressure cooker can be fed with different kind of biomass. And after the reaction, which usually a very short reaction, minutes or few hours, we end up with different phases, gas phase, crude oil, and hydrochar. Osama, after me, will talk about the hydrochar. But now, at this talk, I want to focus on the processed water. And this is important to me because I feel like it's the bottleneck of the technology. Because if you add water or if you use water as a solvent, and at the end of the day you have, let's say, energy product and still some water, if this water will not have any application or useful application, the whole technology cannot be, will not be adopted, will not be implemented. Remember, we don't want to move the problem forward. We want to, if in terms of, let's say, wastewater treatment plant, we need to develop a technology that will solve the problem, in this case, wastewater or biosolid treatment. So I will not talk about crude oil or hydrochar. Amit also worked on that with many students. Some of them are here, I guess. And I don't want to talk about that today. But I do want to say that in terms of energy balance, whatsoever, there is kind of water. Like here, it's the energy balance, different temperature, and each line is like the energy balance in percentage. So you see that for the 300 C reaction, we have these amounts of energy in oil and char, and 5% is water. If we go lower in the temperature, 250 or 200 C, we will find ourselves with more residual carbon in the water phase. In the processed water, ends up with some energy, unuseful energy. So, as I mentioned, 10 to 15% of the carbon still retains in the processed water. This is from Osama's work. We have different temperature, different residence time, and you see here the percentage of carbon in the hydrochar, the solid phase. You'll hear about it in a few minutes, and the processed water. So we have some carbon there, and we do need to treat it. This work was done by Rubia. She was a postdoc in my group. And she wanted to look at the digestibility of this processed water in anaerobic digestion. So the rationale was that, okay, we have a wastewater treatment plant. The wastewater treatment plant has already an anaerobic digestion system, at least the wastewater treatment plant in Israel, based on the activated sludge process. And if we have like if we'll have a hydrothermal compartment in the wastewater treatment plant, then we will harvest some energy as hydrochar. We will still have some processed water. Maybe we can use the processed water and try to make biogas out of it in the anaerobic digestion. So we took some processed water from different reaction conditions, and we actually checked in vitro the biomethane potential, the digestibility of those processed water at the optimal condition, and we saw a very important pattern. That the higher the severity of the reaction, and severity is a combination of temperature and time. So here is like the low temperature, 200 Celsius, 200 C, and short residence time, 30 minutes. And this is the highest severity in 2 hours residence time and 300 C. So you see how the digestibility of the water going like it become much lower, and we see it in the time affected and temperature affected, and end up with the very low digested process. Low digested liquid coming from the 300 C. That means that if we want to make an energy balance, we'll get a much lower energy return from the 300 C. Perhaps we'll have more from hydrochar here, but we will have much less from the biogas, from the anaerobic digestion, and vice versa. If we have a 200 C, we have a processed water that is much more digestible, and that's from the energy perspective. But that also, we have some question regarding, okay, we have some kind of toxicity here. This is toxicity pattern. Something here is toxic to those microorganisms, to the anaerobes microorganisms. So perhaps we may use this toxicity for other reasons. So toxicity is not necessarily a bad thing, at least not in agriculture when we are trying to kill some organism. In this case, we try to use those processed water as an alternative herbicide. So we collaborate with weed scientists, Maor, and we choose to, and we check those processed water as their, how they inhibit germination of different species. And the pattern was actually similar for some species, similar to the microorganisms, but much different behavior to other species. So here you see wheat as a model for a crop, and two weeds. Kres, it's in Hebrew shachlaim, rashad, if you know what I mean. It's a winter type of weed, And this is amaranthus, yarbuz, a summer type of weed, and the behavior of those weed to the different dose of the processed water. Again, the panel works similar to before, 30 minutes, 60 minutes, and 120 minutes, and the y-axis is the temperature. So you see the totally different behavior of the crop if you compare it to the weed. And that's actually very important if we want to actually use it as a natural herbicide. And here we saw also that the fact that even the germinated, this data is just germination, percentage of germination. But here we also saw that even the germinated seeds did not elongate, at least not as the control. And that was also, and this is also very important from the agronomic perspective. So we saw that the wheat behaves actually pretty much, much better than the weed, and also had much better germination percentage. And also we wanted to see, okay, we have that. We want to know what kind of chemicals, or at least what families are we talking about. And in the GCMS, we found again and again similar molecules from the pyrazine families, and that actually makes a lot of sense since we have amino acid and the monosaccharide coming together, condensed and closing this ring, and this actually convinced us that might be one of the mechanisms in the hydrothermal part, or in the hydrothermal reaction, makes this processed water relevant to weed control. Another point of view, and this is the last story for today, was done with the collaboration of Aroma Coffee, or Aroma Cafe. It's a very large company in Israel for coffee, and here we wanted to see whether we can use the spent coffee ground as a feedstock for interesting chemicals, and this was done by another postdoc, Damu. And Damu took the spent coffee grounds from the cafeteria, and I don't have a methodology slide, I guess, but what he did is he ran it in the reactor, had the processed water develop, and irrigated plants after those plants were contaminated artificially with pathogens, like spiking. And he saw that here we saw a very interesting pattern, that the low temperature, the low severity processed water, actually the plants that were, again, infected by those pathogens. This is botrytis, gray mold, I think the disease, and this is kimachon, שכחתי את השם, powdery mold disease, and this is a bacterial infectious. And look at the significant effect of the plant that were pre-treated with our processed water, comparing to the control, means that this processed water somehow induced resistance against pathogen. Remember, this is drenching. The leaves did not see any of the processed water. Everything is from the root zone. So the plant suck all this chemical. And for me, I'm not a plant scientist. It was very new, and I was very surprised by the ability of the plant to become resistant to some biology based on the chemical in the root zone. And you see here that the infectious on the leaves were much lower. The disease was much lower when those plants was irrigated with the processed water. That was like. I'll move very fast. We saw that it also affects the plant yield and the plant other plant physiology parameters. We moved it to the greenhouse, so that's maybe something similar to the previous talk, that we are not stuck in the lab. We actually use. Thanks to Amit reactor here, we were able to produce enough processed water to irrigate the greenhouse in Prigan. It's like next to Gaza, and we saw actually very, very promising data for the farmer. So just a quick summary. Clean technology are required for renewable resource. That's why we're here. I showed the hydrothermal technology and how it can be implemented to sewage sludge. Specifically, we saw negative correlation between methane potential and the severity, and positive correlation between the herbicidal activity and the severity, while the low severity can be also used for other purposes, such as biostimulant or induced resistance of plant. And from a circular economy perspective, the more application we have for this processed water, the better and the faster this technology could be adopted. Thank you.
Question? I'll ask you because it's interesting to me. Can you spray
Yes.
this solution rather than
Yes.
irrigating?
Yes, and we did, and we tried that, and it was successful. Yeah, it's a matter. That's a agrotechnical decision. Sometimes the farmer will prefer to spray. Sometimes he or she will prefer to irrigate it with in the line.
With the tree.
Yeah.
Okay. Thank you, Roy. Okay. Last but not least, a PhD student of Roy, Osama, will continue and talk about the solid phase from the hydrothermal treatment. In this last presentation, and Osama will talk about hydrothermal carbonization of waste-activated sludge, a new technology to recover nutrients and address food insecurity.
Thank you, Amit. Good afternoon, good evening, everyone. So today I will present in front of you my part of my master's and PhD degree work in Dr. Roy's lab. So for the beginning, I will talk about the increase in the total population of the global population increase, and how it will affect other factors like environmental burdens, waste generation, food demand, and water demand. So in the year of 2050, the global population will reach around 9 billion people, while it can cause a global increase in global food demand. It will be doubled as it predicted in this year, and it will increase by 100 to 110%. The phosphorus demand for different uses, one of them is for agriculture, also will increase up to 86%, and the municipal waste generation is expected to increase by 70%. I talked about P demand, food demand, and waste generation. If we look at the left figure, we can see that the P production will reach a peak in around 20 years, and then we will see a decrease in the P production. So it's a kind of concern or problem that we will have an imbalance between the supply and the demand. But if we look to the right figure, we can see a kind of a circular economy model in any town, city, or even country. If we look at the wastewater as a sustainable resource rich in organic carbon, minerals, and macronutrients, as Kader, we can say that the production of the wastewater is ranged between 360 and 380 billion cubic meters per year. It will reach the wastewater treatment plant. In different treatment or applications, we can treat this wastewater and gain a valuable product that we can again use in the agriculture or in the industry. So if we adopt the waste to resource perspective, we can take a low-grade waste into a higher grade product. As I said, a wastewater treatment plant can treat the sewage that reach into the wastewater in different ways and methods. One of them is the waste activated sludge, the product that we get after the ASP, the activated sludge process. So what we can do with the forward with that byproduct, as we all know, the sludge is the byproduct of the wastewater treatment process. It's rich with the organic carbon, minerals, and bacteria, microorganisms, and pathogens. So if we leave it to stand, maybe it have a negative effects on the environment, like it can emit undesired gases such as greenhouse gases. It can also release bad odors. The leach outs from the sludge can reach the groundwater and also contaminate it. But on the other hand, the waste activated sludge can be a material that can help in resource recovery, and it may save money and potentially replace finite resources such as energy and nutrients. In some literature, I see that sludge contains enough phosphorus that could theoretically replace 40 to 50% of the annually applied mineral P as a fertilizer in the agriculture. So as Roy talked about the hydrothermal carbonization technology, he talked about the liquid phase that we get after the hydrothermal carbonization treatment, the processed water, and its uses. But my work In my work, I actually focused on the solid phase, the byproduct that we get after the hydrothermal carbonization, which is called hydrochar, and it's a solid material rich in carbon and other elements. In my work, actually, I worked more on the agricultural benefits from these materials and the different applications in the agriculture. So our main question was, can we use this material as a soil amendment? How the reaction, like the quality of the material is important in terms to use it in the agriculture. So how the reaction conditions can affect the hydrochar quality and yield, how the reaction conditions, time and temperature can affect the phosphorus recovery and the phosphorus bioavailability, and how the use of the hydrochar can affect the plant biomass and the phosphorus uptake by the plant. So when we talk recovery, the most important thing is to know how we get after we finish the reaction. We start, let's say, with 15 grams, how we will get after finishing the hydrothermal reaction. So in this figure, we can see the hydrochar in yield in different conditions, 9 conditions. The graph is divided into 3 groups of time, 30, 60, and 120 minutes. And in each time, we have 3 different temperatures, 250, and 300 C. We can, from this figure, we can see a significant difference between the treatments, but only between the 200C and 300C in the group of 30 minutes. But we can see a clear trend of decreasing in the hydrochar yield after the hydrothermal process in all the treatments, which can confirm the dependence of the yield of the hydrochar in both temperature and time. Also, we can see a nice recovery values that range between 50 to 70% in all treatments. So I talked about the P demand and the way to find alternative to the current budgets of P. So in this sheet, we can see the P recovery values in the different treatments that I showed before under the same conditions. We can see a high recovery rates of phosphorus after the hydrothermal carbonization treatment, between 80 to 100%. These nice results can say important thing that during the hydrothermal carbonization, it seems that most of the phosphorus is precipitated or settled down on the hydrochar's surface. But our question is it available to the plants? It's fine. Most of the phosphorus is recovered, but what we can do if it's not available to the plants? So for answering this question, we did a chemical test, a sequential extraction for the phosphorus according to Headley method. In this method, we tried to see the different fractions of the phosphorus that are soluble in different solutions, water, bicarbonate, NaOH, and an acid HCl. The most or the immediate bioavailable phosphorus is found in the bicarbonate and the H2O extracted fraction. We can see clearly from these pie charts that when we increasing the severity, which is the time and temperature of the reaction, we can see that the portion of the immediate available P in the hydrochar is getting decreased. Like when we look at the 200C 30 minutes and the 300C 120 minutes, the big fraction is for the extracted P by the HCl acid. After that, we proceed into a biological test, greenhouse experiment. This experiment was done in a collaboration with Dr. Ronen. There we found like something opposite to what we see in the chemical test. Here we can see a greenhouse test for corns under different treatments, control treatment without phosphorus, sewage sludge ash, and triple superphosphate chemical commercial fertilizer as a reference treatments, and the corns that they grow with by adding the hydrochar. We can see that the corn ears here show like we can see that they are visually more healthy, but even we are looking to the results, we can see that the hydrochar shows a significant difference to sewage sludge ash and the control test, and even it's not so significant to the TSP. It shows a higher values. Showed a higher values. So the question, we had another question that how come low-grade phosphorus behaves as as chemical fertilizer? So here we assume and we find in the literature that under the high pressure and higher temperatures reactions like the thermochemical, like the hydrothermal carbonization treatment, the most dominant fraction of phosphorus is a polyphosphate. We assume that the polyphosphate may be created under these conditions, and we decided after that so we talked about something that can substitute a found and usable fertilizer that is used for years, and it shows a good and defined results. So in terms to make this product more in terms of commercial product, so we decided to add an economic and environmental layer to our research. So the further work will be about doing a whole life cycle assessment and a whole TEA techno-economic analysis to the process itself and the product process, like to the hydrochar. Finally, I want to summarize and say that since HTC is considered an innovative technology, this work examines the potential of this process to be an integral part in sludge management and wastewater treatment plants. Hydrochar can be used as a soil additive since it contains phosphorus, nitrogen and other micronutrients. Nutrients recovery is directly related to the reactions condition. And although chemical analysis suggests lower availability of hydrochar P, greenhouse experiment demonstrated a promising pattern comparing to chemical fertilizer. And based on the LCATA framework that we will do, it will be developed to optimize the process conditions and maximize resource recovery from waste activated sludge. Thank you.
Question? I have a question.
Okay.
Do you think it's only the P that is important? Why is your corn grow way better than the TSP process?
No, not only the P. We saw that, I didn't show the results, but we showed that in the hydrochar we got a high intensification for micronutrients. And the hydrochar has a high surface area and high organic content that can improve the soil to capture or to maybe water or ammonia or other things. But other thing that we saw that the nitrogen is getting decreased when we increase the reaction severity.
Okay.
Thank you. Yeah.
Okay, so we're late. Thank you.
Thank you.